Bueno, mentre, si voleu, presentem en Marc, que molta gent ja el coneix, crec que és de, de les persones que més que tothom coneix. Doncs, bueno, perquè no sapigueu, en Marc va estudiar bioquímica aquí a la Universitat Autònoma de Barcelona i després, el 2017, va entrar a l'ICMAB, primer a fer el màster amb la Imma Ratera i la Judit Guaix, i després que el màster era d'Advanced Nanoscience and Nanotechnology, i després va començar la tesi. Durant la tesi va estudiar cèl·lules en 3D, amb estructures tridimensionals i desenvolupa sobretot per teràpies per càncer i d'immunoteràpia. Estava becat per l'FPI, tenia una beca i estava a la supervisió. La tesi al final va augmentar una supervisora i va ser la professora Nora, la Imma i la Judit Guaix. O sigui, 3. Va tenir tot un molt suport de tothom. Va rebre el doctorat al març del 2023, per tant, recentment, i després el vam incorporar, el vam cridar per veure si es volia fer càrrec del servei, que ara us explicarà que el servei hem tingut anat renovant i ell portarà una part de rejuveniment i reestructuració del servei. Per tant, Marc, dada.
En anglès, no? La presentació. Bueno, suposo que millor. Sí. Sí. Okay, thank you all for coming, all the known faces, since I'm from ICMAB. This is a short presentation that I have prepared to present to you, for those who don't know, and almost all of you already know, to present you the bioservice that we have at ICMAB, what we can do, how you can work with us, and also some considerations for the people working in material sciences that want to start working with biological agents. And there are always some kinds of hints or things that you have to take in mind when you have to start working with biological agents. So first, the bioservice is a relatively young service at the ICMAB. It was started at 2020 to aid ICMAB researchers in experiments with biological agents, mainly mammal cell culture and bacteria. It had an establishment phase from 2020 to 2023, thanks to an agreement with the ICN2, because we are using their infrastructure, their biolab facility. And then on 2023, as Anna said, they hired me to try to give an impulse to the service. And now, during 2024, we are in a strengthening phase of the service. We have revised the services that we provide. We have applied to equipment calls. And also, recently, we have acquired a working space in ICMAB that we did not have previously. And looking until 2025, we want to improve the capabilities of the service in order to better support all researchers at ICMAB. We are two people working at the service, the scientific coordinator, Dr. Ana Romain, who has very kindly presented me, and myself. And so, working with biological agents. What is a biological agent? From the point of view of Spanish law, at least, it's an agent able to originate any type of infection, allergy, or toxicity. And this also applies to both alive biological agents and products derived from them, as toxins, for example. Some examples of biological agents are microorganisms, bacteria and virus, cell cultures, and also some human endoparasites, as protozoa or helminths. And in fact, biological agents can be found anywhere, even in your fridge. For example, the mold that you can have in your fridge or any bacteria that can grow. However, not all biological agents are the same. In fact, depending on the risk they pose to human health, they can be classified in four risk groups. Going from the group 1, which are agents that have low probability of being pathogenic to human beings, that are not propagated in collectivity, and in which treatment is usually not necessary, all the way to group 4, which cause severe disease, can be propagated very easily, and that may not have any known prophylaxis or treatment. do not be preoccupied by this, because at ICMAB, we work only up to group 2. And in order to work with these kinds of agents, to deliberately work with this, not with the ones that you may find everywhere in your life, you need facilities that have a biosafety level. And this is that this kind of levels of safety measures that you can adopt to work with these 4 groups, and usually by increasing the type of group risk of biological agent that you are working with, you need stricter safety measures. However, as I said, at the bioservice, we work only at this level up to BSL-2, which are mainly in BSL-1, we have some mouse cell lines, some non-pathogenic bacteria, and then in BSL-2, it's recommended for working with all human cell lines, all the cells from humans, and also all the cells from non-human primates, and also some clinically relevant bacteria. So, as I said, we do not have a currently operative laboratory for working with biological agents at ICMAB, and we use the agreement that we have with the ICN2, our neighboring institute, in order to use their biolab facility, which is a BSL-2 laboratory. So here we can work with human cell lines and clinically relevant bacteria. In this laboratory, we have access to all the necessary equipment to work and cryopreserve these cell lines and bacteria. However, we are subject to the availability of their schedule. They have an internal booking calendar, and we have to depend on that in order to work at the ICN2. So how can the bioservice support your research? As I said, it was started to support all the researchers at ICMAB in their biological experiments. And the idea is that we provide a la carte experiments with biological agents according to the needs of your users. So you come to us with, I have an idea, I want to do this, I want to try that. And all together, we can design an experiment and execute it. And as I said, we support all users independently of their experience working with biological agents. So we can help you from just giving advice to try to see if an experiment makes sense or some considerations that you need. We can also execute the experiments, and I can also visit facilities in order to carry out these experiments. For example, in a previous experiment with Rosario, I had to visit the UAV microscopy service in order to bring some cell to confocal microscopy in order to sort of direct the experiment, talk with the technician. All that you need, we can also do this way. And also we can help you in the analysis of experiments. If you are inexperienced analyzing data, for example, for site availability experiments, we can also provide this expertise. Now, how we work, how can you request a service? So the first thing is to reach out, to just send an email to me with a brief explanation of what you need, of what you want to discuss, and then we meet, preferably if it's with a student, also with the PI present, in order so we can all agree on what needs to be done. And then we prepare a quotation, because as I said, we are a la carte service, so we need to design each experiment and each quotation independently, and then we perform the experiment on the agreed upon date. We can bring visitors. When I go to work to the ICN2, we can bring visitors, your students or whatever who needs to attend. This is allowed for the ICN2. However, if you want to do the experiments by yourselves, you will need to be self-users. And this is a bit more complicated because, as I said, we depend on our agreement with the ICN2 to access this facility. And in order to be a self-user, to access the ICN2 whenever you want, you need the access credentials of ICN2. And this means to carry out all their security tests and that they register your fingerprint and that you make an agreement between ICMAB and ICN2. So this is something that we can do, but it's a bit more complicated. And also, when would be interesting to do this kind of self-user agreement, if you have a very high workload of experiments, so maybe if I cannot do all the work, you may need to do it yourself, and also if you need to work independent of me, because it's not the same for me to bring a student, for example, to teach him something, or for example, you that you have already your project, you know how to do everything, and you don't need me for anything. So it could also be beneficial for you to do it this way. But this, since I said this requires an agreement and some credentials with the ICN2, this is best done for long-staying users, such as PhD students or postdocs. For example, for master students that are here for a very short time, it's not feasible to give them this access to the ICN2. And now, yeah, the current capabilities of the bioservice, as I said, it is a BCL2 laboratory infrastructure, meaning that we can maintain and cryopreserve mammal cell lines and bacterial strains. We have in the NeoLabs folder a list of the bacteria and cell lines that we have. And so the typical experiments that we do is cell viability assays in order to see how healthy cells are when exposed to an agent or a material. And we can also do antibacterial activity assays, for example, this diffusion assay or minimum inhibitory concentration. And one of the good things of working at ICN is that they are very flexible. They tell us that we can bring devices or specific setups inside ICN2. If you need to test, I don't know, a specific device or equipment, it can be a real bond to bring this sort of setup inside the ICN2 laboratory. And so inside the ICN2, we have accessible several equipments for the routinary tasks that we can do there. One of them is the microplate reader, which is an equipment that can read absorbance, fluorescence, or luminescence in 96 well plates. This is very useful for having a lot of samples at the same time. Just information, if you do not know, at ICMAB, we have an equipment like this in the soft service. It does not have luminescence, but you have it accessible also at ICMAB. And then in ICN2, we also have access to an inverted fluorescence microscope that can be useful if you need to stain with fluorescence some cell cultures in order to observe them in this microscope. As future perspectives for the service, as I said before, previously we did not have a space here at ICMAB to work with biological agents, but Direction very kindly has provided us with a shared space in the laboratory 230 that we are still, we just received this shared space, so we are working to condition it to eventually be able to work in this laboratory at BCL2. So for now, we cannot just do our activity here at ICMAB. We still will have to do everything at ICN2 until this laboratory is ready to work. And then recently, thanks to the FAST 2024 call, the BioService has recently bought a very nice inverted fluorescence microscope with a very powerful camera that when we receive this equipment, we expect it to be available since January 2025, hopefully. But when we receive this experiment, we will make an announcement for all the users and all the people at ICMAB in order to know exactly what we can, or what we'll be able to do with this equipment. And so now moving to a more didactic part of the presentation, which I think it's like the most interesting, the one that makes you think a bit, is some considerations before going to work in vitro. So usually at ICMAB, we are a materials science institute, so we have all kinds of different materials that we work with. And when you produce your material, maybe you can have it suspended or solubilized in a very simple medium. It's all perfect and very nice. But when you go to biological experiments, the medium tends to get more complex because your material that is very happy in this very simple medium has to coexist with a lot of other species or maybe another kind of medium that may not be very beneficial for its activity. So it's very important for you to check the stability of your material in this kind of medium and also if it can exert its function properly. So I have put here a very brief list of some of the typical conditions that you can find in this kind of experiments in order to think or test if your material is compatible with these conditions. For example, the aqueous medium. I have found many times working at ICMAB that we have materials that are very insoluble. And when we have to go to water-based mediums, which is completely necessary for biological agents, well, maybe you have problems. How you solubilize your material? Is it even stable in this kind of medium? also the range of temperature. This is not, I would say this is a very mild temperature, but you have to take into account that your material will go into physiological temperature. Also pH, typically it's neutral, but depending on the kind of assay, it could go either acid or basic. And also the ion concentration. You cannot have cells in just water, they will die by osmotic shock. So you have to take into account that your material will also be exposed to a certain ion concentration. And then proteins. If you have not heard about protein corona, this is a phenomenon that happens usually when injecting nanomaterials inside the circulatory blood flow. The blood is full of proteins, and these proteins have different affinities, but they tend to be, speaking roughly, sticky. So these proteins can organize on top of your material, and maybe this hinders the stability of your material, or it hinders its function. So this is something that you have to take into account before going to these kind of experiments. So my recommendation for you is to test the properties of your material in the presence of these conditions before going to work with biological agents, because maybe it does not make sense. Maybe your material is not stable, it cannot work. And by doing this, it's much more cheaper than working directly with biological agents to not have any result and to not know why. Also, before going in vitro, you have to ask yourself, is your material sterile? Because when working with cell culture or bacteria, you have to work with them in isolation from other kinds of biological agents, because contamination, usually by bacteria, can affect the reliability of your in vitro experiments. It's not the same having your cell culture happy, so to speak, than have it contaminated by some bacteria or some fungi. So you need to think about how you can sterilize your materials. Here I have put some examples for you. One of the most typically used, if your material is resistant enough, is the steam autoclave. This is used to sterilize the plastic and metal material that we use when working with biological agents. And this uses steam at very high temperature in order to kill everything. This is very effective, but as I said, maybe its temperature is too high for your material. We can also try UV light irradiation, because UV light is very energetic and can damage the DNA of bacteria and cells. So this is a way, by exposing your material or whatever to this UV light, you can disinfect also the surface. And also, a milder approach, if your material is soluble or can be suspended and its size is smaller than this cutoff, is syringe filtration. Through this way, you just load your material in a syringe and pass it through a filter that has a size that entraps most bacteria that you can find contaminating your samples. And now some examples of the kind of experiments that we can do in the service. One of the most typical ones is the cell viability assay, which is an experiment by which you expose your cell culture to some compound or material, and then you assess the metabolic activity of your cells, and is typically used with commercial kits that are very standardized, which are, for example, the MTT, XTT, or Alamar Blue. And the process, very briefly, is that you expose your cell culture to some material, compound, or whatever you have, and after some time, maybe your cells are the same, maybe there is no effect on your culture, but sometimes this material, if it's toxic in any way to cells, it can cause cell death or stasis. So after this exposition, we used these reagents from these commercial kits that are processed by metabolically active cells to yield a product which is colored or fluorescent, anything that can be detected by this kind of equipment that I showed you before. And so in this case where we had no effect, the higher number of metabolically active cells, it's represented by this higher signal. However, in this other case where we have dead cells, there is a low amount of viable cells, so the signal will be lower. And thus, comparing these results with some controls that you include in your experiment, you can see if your materials are damaging the cells. And this can be interesting when you when you test this, because depending on the kind of material that you are working with, maybe you expect or want to look for different responses. This is made up data. This is not real. This is just to show you a representation of the kind of results that you can get. Maybe you have an agent that you want it to be diagnostic. So you want this agent to not exert any pressure on the cells, to not kill anything, so to speak. So maybe you see here that these low concentrations of the theoretical concentrations of your agent, maybe it's not affecting much the viability of cells in comparison to the control at 100% viability. But for sure, there can be an effect of the concentration. There are a lot of compounds and molecules that at low concentrations are not toxic to cells, but by increasing them, it can become toxic to your cells. So this can also give you information about the range of concentration that is tolerated by cells. In comparison, when you have a cytotoxic agent, something that actively kills cells, you can see results as this one, in which a low concentration of your compound already kills the cells. And this is interesting because you can calculate kinetic properties of this kind of experiments. So through this kind of cell viability assay, you can see the range of concentrations, which is compatible for your material with the cells, or you can also try to have a more quantitative look on the effect, on the active cytotoxic effect of your material or your compound over cells. And now moving to bacteria, a very easy to do assay is the disk diffusion assay, which is typically used in the clinics to see the susceptibility of certain bacteria to a range of antibiotics, but we can use it too to see if the bacteria are susceptible to some bactericidal compounds that we can develop at our institute. And in a very straightforward way, we load our bactericidal compounds on cellulose discs that then we put in agar petri dishes inoculated with bacteria. And after some time, we can see a confluent bacterial growth. And if our compound has an antibacterial activity and the bacteria are susceptible to this activity, we can see around the cellulose disc a zone of inhibition where the diffusion of this compound has prevented the growth of bacteria. This is a photo, so you can see more clearly what I see here. It's I don't know what bacteria this is, but this is different discs with different antibiotics. And you can see that for some cases, there is a susceptibility, a prevention of the growth of bacteria. And in other cases, there is no prevention. However, this is a screening technique that can be very useful for us in order to see very quickly if, if our compounds, your materials, are preventing the growth of bacteria. This is also to say that this depends on the ability of your compound to diffuse through the agar. So this is not compatible with all kinds of materials. And also, if you want to look to a more quantitative way of having the activity, the antibacterial activity of your material or compound versus bacteria, we can also do the minimum inhibitory concentration assay, in which here, simplified, we have some tubes in which we have diluted twofold the concentration of an antibacterial agent. And then in all these tubes, we inoculate them with bacteria. And after 24 hours or overnight incubation, we can see that in some tubes, there is a growth of bacteria. This is visually seen because you see that the growth medium that is clear gets turbid. You see that something has grown inside. So here, for example, in this example, we could see that in these tubes, after 24 hours, we have a bacterial growth, meaning that for this experiment that we did, this would be the concentration of our antibacterial agent that have prevented the visual growth of our bacteria. And so this assay, which is also very simple to do, can give us more quantitative data on the antibacterial activity of our material. And so this is more or less everything that I wanted to show you, but the take-home message that I want you to bring with yourself from this presentation is that my door is open for you to come to me and talk and discuss, because I understand that in a material science institute, not everyone is exposed to the intricacies of working with cells and bacteria. And I feel like the value that we provide with a la carte experiments and advice and everything can be very valuable for you in order to design your experiments. And so thank you very much. And if you have any comments or questions, I'll be happy to answer. S'ha apagat.
No sé, l'he posat malament. No funciona, ja no funcionava. Bueno, moltes gràcies, Marc. Thank you very much. Is there any question for Marc? Come on, please.
Almost everyone knows me already,
Yes.
so. Is there any question from the people looking online?
So we wanted to encourage you to use the service now that we have a technician that knows all about it. Maybe you can comment. I think we could also comment which percentage now is more bacteria experiments, cells experiments, and if also users.
Yeah, right now, our most the users that use more the service are the N&N group and the nanomol bio group. We have done some singular experiments for other groups, but mainly our users are N&N nanomol. And at first we did more cell culture experiments, but right now it's more bacteria, it's more microbiology.
No idea what the biology but can you take your samples of some bacteria the labs outside the to other lab in ICMAB?
It depends. I mean, if they are group 1, they should be brought to a laboratory that is prepared to work with risk group 1 and biological agents. And the same for group 2. You should not bring unless it's, I don't know, you have sealed your sample and just for a quick measurements. Maybe you can do this, but in principle, everything should be done at the laboratory in the ACN2 or in the facilities, for example, the ones at SCAC or the microscopy service.
I understand that there is some people that bring you samples to AFM.
Yeah.
So, for instance, how it would work is there is a procedure to fix the cells and then
Yeah.
preserve the structure of the cell and then cells are dead. And you have to think that most of our experiments are in liquid. So it's not a cell cannot be on air, so it's not very easy. So it's very difficult to propagate. It's very safe on that. So for that reason, also the joke on the sometimes in our fridge is worse than what we have in the lab. So there is this it's bio, it's very dangerous. Well, be careful on your fridge sometimes because it's worse than what we have in our lab. So that for that reason, Mark added this slide. And then you can just bring your samples that are either figs or not, but in nano, for instance, probably you will see Mark or some students that go to the microplate either. But unless it's completely still, then there is something not going to happen. Sometimes there are other solvents that they are maybe they can be vaporized or they can have vapors that are worse.
Yeah. Yeah, it depends. For example, as she said, if the cells are fixed, their structures are preserved, but they are not alive anymore. So this is not dangerous, for example, to bring around. And a lot of samples for microscopy, for example, if you do not want to see alive cells, you fix them and then you have them preserved and you can bring them anywhere you want and it's not dangerous or anything. But yeah, it depends on the technique that you want to do. I don't think so. No, it requires very specific infrastructure. And at ICMAB, I don't think we have the expertise or the need to work with.
You can comment the ICN2. It's a BL2, we could say BL2 plus. They have the room actually has the top part has an extra filter ventilation that is constructed in a specific way to actually have an extra ventilation. So imagine you're at the ICMAB, a new construction with the ventilation. So, and also we don't have the resources. We are a materials institute that we want to test. We think that this is going to be exploited and trying to improve our services, to test our material, to then maybe go to animals or a further experiment, but to do initial experiment or some kind of experiments here in-house and then maybe start with a collaboration. Because sometimes you cannot interact with doctors or very advanced biological labs saying, this is my material, I don't know anything. And they say, well, you don't know anything, first you should know something, right? So this is what we can actually do in that place.
And the laboratory 230, is it ready to start work or?
For us, no, because we have everything at the ICM2. We have to, it's a bit complicated. Yeah, because there are still some equipments from other services that still cannot be moved because they need specific, they cannot be moved right now. So until we move those and we want to clean the laboratory, we also have to bring a biosafety cabinet that Nanomol has donated to the service. We still have to move it, so it's a bit, and it requires a bit of investment.
Both laboratories here and...
Yes, for now, yes. However, I expect for some months, maybe, to all the work still be done at the ICN2. Hopefully, eventually, we'll be able to do everything here, but for now, it's not something that we'll be able to do at the moment. Eventually.
They work a lot in many groups working, but they have a specific group that they nice. And then here also there are other experiments that we have. We can do protein quantification or some preparation. Some group may need some sterilization. The sterilization that Mark mentioned, we have it here at the ICMAB. So we can mention that we have an place at the ICMAB, there is UV, or we have syringes. Because there are other methods. For instance, it's gamma rays, but we don't have it here. But those ones that we presented, we have it at the ICMAB. So we can sterilize it within those systems.
Very nice. Very nice to see how things advancing the detection of specific just one specific question regarding the cell lines that you have. I know that you are little by little like expanding the inventory of cell lines. Do you have a clear list of all
the list is in the Neolabs laboratory. However, we do not have really the ability to buy new cell lines because these are very expensive. And since we are a service that is starting, we do not have a lot of money. There are some lines, that we have a list in the Neolabs, or we can talk and I can show it to you. However, if you need anything that it's not in our list, for now, the service cannot assume the purchase of these cell lines, so it depends on the users. We can maintain them we can preserve them in nitrogen and whatever you need, but the buying new cell lines, it's too expensive for us at the moment. But we have some small variety, so.
We have been able to keep some bios, and we can actually do it. That's true that from our portfolio cells, you don't have even bacteria. The users can buy it, and then we ask the users if they don't mind to keep a vial, and then we can use it for other experiments. But right now, it's what is on the
it's already very long, but there are very different kind of cells. And that is that I saw sometimes was okay.
you know where it's marked.
and you can come.
My door is open.
Yes. And encourage you to actually be able to use it and test and seek for the advice of Mark for your experiments. Okay, thank you very much.
Thank you.